Can you hear me in back, Rachel? Does the sound okay? Okay. I'm Eric von Wettberg. I'm the moderator for this session in population genetics and the first speaker. I'll go ahead and get started since I'm a minute or so late. I'm going to talk this morning about germplasm collections for chickpea and their utility with expansion for addressing evolutionary questions. So we know that since the beginning of evolutionary biology, domestication has been of interest to evolutionary biologists. And we can think of this as having two components. One is our ancestors selecting quite strongly on a handful of traits that are essential to convert a wild species into an agricultural crop. The second component is more of a stochastic process, inadvertent consequences of domestication that may have very strong effects on the traits of our cultivated species. I'm going to look at this morning in chickpea. For most of us from the West, this is chickpea. It's in the center. It's the source of hummus. However, it's a far more important crop than just hummus. It's the primary source of protein for about 15% of the world's population, primarily in South Asia and East Africa, as flour, as a whole or split pea, fresh vegetable, or a beer nut. It is surrounded in this image by seeds of other annual cicer species. These are all wild species. This creature here is Cicer reticulatum, the immediate wild relative of chickpea. It's interfertile with cultivated chickpea. This down here is Cicer achinospermum, which is sister to these two species and only partially compatible. The rest are fully incompatible with cultivated chickpea. Chickpea is a Neolithic Middle Eastern domesticate, so along with wheat, barley, lentil, pea, it was domesticated in the northern Fertile Crescent 9,000 to 10,000 years ago. Unlike the other Middle Eastern domesticates, the wild relative has an extremely restricted geographic range. It's restricted to a handful of predominantly Kurdish provinces in southeastern Turkey. By about 9,500 years ago, large seeded chickpeas turn up in archaeological remains outside of Aleppo and in other areas, and within a few hundred years are well beyond the known native range of its compatible wild relatives. By about 6,500 years ago, it had moved to the Indus Valley in South Asia, where it's been culturally significant since then. To domesticate chickpea, our ancestors in the Middle East must have selected on a few traits. One is non-shattering seed pods, as with many other annual seed crops, large seededness, erect growth form, and unlike some of these other Mediterranean annual crops, our ancestors likely selected very quickly on early flowering. This is because chickpea is sown as a spring crop in temperate environments after winter rains in the Mediterranean, or in South Asia and East Africa after the monsoons. This allows it to avoid Ascochyta blight or Fusarium wilt fungi. We can take approaches used in other crops that have lagged in an orphan crop like chickpea to map the genetic basis of these. We've done this in flowering time, where if planted at the same time in the same field, a cultivated parent and a wild parent flower at different times. And the F5 offspring from this cross are roughly bimodal in when they flower, some with a flowering time around that of the cultivated parent, and some with a more delayed flowering time, so with some transgressive segregation. And we've used RAD-seq to map QTL for this. This is an increasingly common method, but in short, we've taken genomic DNA, done a double restriction digest, Illumina sequenced the pieces, so this is dd-rad, and we've assembled it against the recently published cultivated chickpea reference genome. And in our hands, this generates a few 10,000 single nucleotide polymorphisms, which are more than adequate to map QTL in a diploid 750 or so megabase genome. In flowering time, we find a single QTL that accounts for about 70% of the variation in flowering time. It's on chickpea's third chromosome. It's a somewhat complicated QTL in that it affects flowering time, but also affects reproduction across nitrogen treatments in a greenhouse. We think there's an ortholog of a well-known flowering time gene underneath this QTL, but we're still verifying it in a second cross and verifying it in which I'll talk about in passing in a few minutes, an expanded collection where we have the capacity to look for selective sweeps around this locus to see if it's indicative of a domestication locus. Some of these other traits have a similar genetic basis, but flowering time in chickpea is particularly interesting because if we go to the field in southeastern Turkey, the wild and the cultivated actually flower at the same time. This picture was taken by me in mid-June in the middle of a wild chickpea population on the side of a Kurdish hillside. Down below is a cultivated chickpea field. They're both flowering at the same time. The wild in this area has a purple flower, and the cultivated is a white form. Despite what Norman Elstrand will tell you, we find a handful of purple individuals at the edge of this cultivated field, and they have an erect, only partially erect form, suggesting that there's some ongoing hybridization that occurs in this area. Because of that, we think early farmers in this region, after domestication, selected for flower color, in part maybe for it's simply its visual appeal, but also in part so that hybrids can be easily recognized. So the wild and something called the Desi market type, this is the most common kind of chickpea in India and Ethiopia where the wild sicers don't occur. These have a dark seed indicating condensed tannins and a purple flower, while the common cultivated type in the Middle East and much of the West has a light colored seed and flower. This is indicative of an anthocyanin condensed tannin pathway mutation. We've been taking the same QTL approach coupled with a candidate gene approach, since this is a pretty clear phenotype, and found the ortholog of Mendel's B locus, a basic helix loop helix transcription factor that affects the expression of both anthocyanins and condensed tannins. And in Kabuli genotypes, we find several mutations that look to knock out its expression. These are two traits that we think our ancestors selected on in chickpea. But there are a number of other traits that may have come under stochastic processes. In annual crops, we often think of a strong domestication bottleneck that removes genetic diversity from first heirloom varieties, as our ancestors select a few individuals that perform well in traditional agricultural settings, and that is then exacerbated in the last century in modern breeding as we intensively select for high-yielding agricultural varieties. This is well documented in crops like maize, wheat, rice, soybean, intensively studied crops with enormous germplasm collections. Chickpea and many other crops are historically orphan crops that receive much less attention. Breeders for these crops in general are very hesitant to use wild material. They've never collected it intensively. And as a consequence, most international germplasm collections only have about 2% wild material. The use of it, if it's used at all, is generally ad hoc. And in many cases, it's missing. I'll describe this further in chickpea, but this is a common problem across many secondary crop species. In peanut, crop domesticated here in Brazil, in the USDA germplasm collection, there's only a single accession of one of the diploid progenitors that's available. If if you want to make any inference about the processes underlying the domestication of peanut, the collection is simply inadequate. In chickpea, there are hundreds of cultivars in international accessions, and with duplicates, you can make that number thousands. But only 18 genetically unique members of the wild relative are present in gene banks. And almost all of them come from a single village. It happens to be a lovely village, but they're all from a single village and the side of the road, and incidentally close to the bar. We've looked at this, I didn't do the collection. We've looked at what diversity is present in the collection, nonetheless, using a subset of SNPs. And we see, if we look at gene diversity or expected heterozygosity, that across all 8 chromosomes listed here, in general, the diversity of the wild is higher than that of the cultivated. But what this means is something for adaptive variation or agricultural performance is something that we're not sure of. I started a minute late, so I'm going to skip through some of our preliminary pilot data on nitrogen uptake, but we have evidence suggesting that there are differences in how both mineral nitrogen and mineral nitrogen in the presence of rhizobia are utilized between wild and relative, between wild chickpea and cultivated chickpea. But if a collection only contains 18 individuals of the wild relative, it's clearly inadequate to make inferences about loss of variation and its impact on traits. So we've set about creating a better comprehensive collection, and we used a brief lull in the 30-year conflict between the Turkish military and the PKK to collect through southeastern Turkey. In doing so, we expanded by an order of magnitude the collection of wild chickpea. In a RADSEEK survey, we find we've greatly increased the amount of diversity present in it. We're still processing this in terms of understanding variation on a genome-wide pattern. So stay tuned, we're going to come back to this. In addition, we're assembling an improved landrace collection. Landraces are underrepresented in general. In particular, we have access to a unique collection assembled by the great Soviet collector Nikolai Vavilov, as well as Turkish and Ethiopian collections. And we're starting to introgress it into cultivated material. So, having started a moment late, I'd like to quickly wrap up and stress that for an orphan crop like chickpea, if we want to make proper inferences about domestication, or if we want to effectively utilize the material, we need comprehensive collections. And I'd like to stop by saying these are only possible through collaboration with a number of breeders. I'd like to thank our support from USAID, NSF, and other sources. And finally, so that I have time for a question, you can tell we have a lot of population genetics to do. I am looking for a postdoc and have opportunities for students. Thank you very much.